Karen. So with that, I'll introduce Harald and to tell us about the end of medicine as we know it. Thanks, Harald.
Thanks, Rob. And now that you mentioned my Australian stations, I just remembered my PhD supervisor was Harry Majewski, the pharmacologist from RMIT. So some may know him actually. Yeah, the end of medicine as we know it. So it's not the end of medicine. It's only the end of medicine as we know it now. And why? It's actually not a little thing. So a lot of futurologists think it's the next big Kondratiev wave. So you may not have heard of Kondratiev wave. These have been the major socioeconomic revolutions in our society. So the invention of the steam engine, actually a response to an energy crisis in the UK, enabled the Industrial Revolution. And the invention of an electric light bulb is not an incremental improvement of a candle, it's something completely new. So all these Kondratiev waves have in common, they respond to a crisis, things that we thought will be there forever, like sailing boats, for instance, commercial sailing boats, will disappear, and things appear that we would never have thought would be possible. And they usually last 40 years. Max Planck has said, every innovation comes one funeral at a time. Because real paradigm shifts, of course, induce huge resistance. So I think we can all agree that the most recent Kondratiev wave was the IT revolution. But we have now the Amazons, Apple, whatever, yeah? So what is the next big thing? And almost all futurologists think it's not renewable energy. That's just a new energy form. It's obvious that we have to use it. It's not Industry 4.0, which is usually doing the same thing, but digitalizing it, making it a little bit more efficient. That's not a paradigm shift. They think it's a complete redefinition of psychosocial health. How we define health, how we preserve health, most importantly, and how we cure. And as I said, they all start with a crisis. So you might say, what is he talking about? Crisis. We have such a wonderful medical system. We can cure so many diseases. Mortality has decreased since the 1900s. Yes, it has. That's a decrease of mortality since 1900. That's the Spanish flu, this little peak there. But if we look, what has been the contribution of preventing and curing infectious diseases? So hygiene, vaccination, and antibiotics. That's the red line. And when we subtract the red line from the black line, this is what the rest of medicine has achieved since 1900. In fact, since a couple of years, the life expectancy in industrial countries is decreasing. So 80% of the costs in our healthcare system are actually only due to about 15 conditions. Diabetes, number one. We have now a diabetes pandemic of about 8–9% of the population. 20% of all adolescents have pre-diabetes already. So it's one of these conditions that contribute to all these chronic diseases. We could prevent them actually by keeping 8 risks and behaviors under control. This lower one, poor standard of care, I think this we can eliminate for countries like Australia. So that are 7. Physical inactivity, not 10,000 steps. You all know that was a joke at the Tokyo Olympic Games because There, the first pedometer was invented, and the Japanese number of 10,000 looks like a running man, so they put this running man on there, and since then everybody thought you have to walk 10,000 steps per day. It has now been investigated about 3,500, 4,000 are sufficient, but a lot of people don't even manage to do that. You know, go from home into the car, up to the office, back to the car, back home to the couch. Smoking is clear. Lack of health screening, mainly men. Men have 5 years shorter life expectancy than women. Poor stress management. We all have stress, but one can learn to manage it. Insufficient sleep, major risk factor for Alzheimer. Excessive alcohol consumption and poor diet, lots of red meat. It's a risk factor for colon cancer and coronary artery disease. So very simple things, but they're not part of our healthcare system. Actually, we have a sick care system. We spend only 1% of our healthcare budget for prevention. We mostly deal with the disease state. So we run things into dysfunction and then we try to repair it. And very often by drugs. About 70% of what a medical doctor on average does, not when you're a neurosurgeon, but in average is prescribing a drug. Then you might say, so what? We have the drugs, we can treat it. So this shows the 10 drugs with the highest turnover in 2015, but in principle it hasn't changed. And the green patient are those who will benefit from these drugs. And the red patients are those who will also receive those drugs according to guidelines, but we know from all regulatory trials, they will not benefit. If they have bad luck, they will actually have the side effects. This is not a conspiracy theory, that's the number needed to treat. How many patients do I need to treat with one condition and one drug so that one patient has a benefit? We have no means in the moment to differentiate between the green and the red patient. There's a site where you can look up, the NNT.com. For every drug, for every therapy, what's the NNT? This has affected the pharma industry. You may have heard that pharma had an exponential increase of costs in the 90s, and that's the blue area. And if you look at the new medical entities that have been approved with all that money, that's the green line, it's more or less stable. So if the costs in an industry explode and the outcome stays the same, I think we can all agree that this industry has a slight problem. And it's not a recent problem. It has been analyzed by Scannell since the 1950s. The number of drugs approved per $1 billion spent is in a constant decline since the 1950s. So obviously, there is a conceptual problem how we develop drugs. It gets harder and harder and harder. And it's not due to tighter regulations post-thalidomide. And it has also not improved with the introduction of biologicals. They almost have the same attrition rate. There is a conceptual problem. And now you get every year less than one drug per $1 billion spent. So in 1 year, it's $2.8 billion to get one drug to the market. In some years, it's $5.4 billion to get one drug onto the market. And what about those drugs that are actually, that make it to the market? It's been analyzed by the German regulators that 2/3 of the drugs that actually make it to the market, when they're tested in real world patients, not in the hand-picked patients of a phase 3 trial, optimal age, no comorbidity, tightly monitored and so forth, there's no benefit remaining in real-world patients. One consequence of that is that pharma has almost retracted from all clinical areas. This shows all medical indications. The size of the bars shows the developments in recent years, and red, the most recent ones. And you see there's not a lot of red on this graph, actually only down there. So pharma is focusing in the moment mainly on immunomodulating drugs, on anti-cancer drugs. You can always bring any anti-cancer drug to the market, even if the gain of life is a few months and so forth. And I think you have read the papers in Science on how many cancer trials have been manipulated, and antiviral drugs. In many areas like cardiovascular, neuro, you heard about the Alzheimer disasters. I think 7 major projects now failed. Silence. But there are many problems that lead to that. The most profound problem, I think, is that we do not understand diseases. What usually happens is that a patient with no symptoms comes to a doctor, and the doctor measures elevated glucose, elevated cholesterol, elevated blood pressure, whatsoever, or comes with a symptom to the doctor, can't breathe well anymore, can't exercise so well anymore. And then the disease is named after the symptom. High blood pressure, asthma, inflammatory bowel disease. Fantastic disease definitions. We describe the symptom in an organ. We have no idea what's causing it. And then if you can't cure something, if you can't repair it, it becomes chronically defect. So the increase of chronic diseases is a capitulation of medicine. We do not understand diseases. We fiddle around with chronic symptomatic therapy with very low precision, and therefore we don't, when we actually measure it, achieve very much. There are lots of reasons. You know, in basic research, completely false incentives in medical research. To make a career, you have to publish a lot, you have to have research income, and with this research income, you publish a lot. So no one measures what actually comes out of all this medical research for patient benefit. It's not a career criterion. But the biggest conceptual error that we all made in medicine, and of course I myself, that we have divided up the human body organ by organ by organ. For every organ, we have a clinic, a clinician, a specialist, and a research discipline. We have neurology, the neurologist, and neuroscience. We have cardiology, the baker, and cardiovascular research. Yeah, so we really think by looking at one organ, whatever we see there is the disease. Whatever the patient has in any other organ is another disease. Has nothing to do with my disease. I refer him to another specialist, let another research discipline deal with it. Nothing to do with my disease in my organ. And if you think already of monogenetic diseases, rare diseases, where one gene is involved, these patients very often have symptoms in 3, 4, 5 organs. This simple example tells you an organ-based approach is wrong. It's understandable, you know, at some stage, the medical knowledge had to be structured somehow. Why not organ? But we have to realize this approach has to end. It's leading nowhere. Or we name a disease after a doctor or a symptom, Alzheimer, Parkinson, and so forth. So enough negativity. Now to the positive side of things. So, and it actually has to start here. This was for me a eureka moment. When I saw this paper by Laszlo Barabashi, he is a physicist, actually, a network scientist. He does all kinds of networks, social networks, whatever networks. And in this paper, he did the network of all human diseases. And the color code is of what I just talked about, you know, this organ-based approach, how to structure medicine. And in this paper, he networked the diseases by shared risk genes. So this gene here is a risk gene in 3 diseases. And if you construct such a network with all diseases and all risk genes, you end up like with this. What becomes apparent is that it's not a homogeneous network, it's actually a scale-free network where the diseases or some diseases cluster together, they form little groups. And you will appreciate that most of these clusters are multicolored. This one, that one, that one, that one, and so forth, which shows again diseases with symptoms in different organs, which we treat by different doctors, which we do research on in different disciplines, genetically belong together. So the only thing that we do now is we turn this around. What we now call diseases, we just call phenotypes. And the diseases we want to extract from these risk genes that keep those clusters together. So obviously, there must be one or more common mechanisms in this cluster, causal molecular common mechanisms that lead then to phenotypes in this group of diseases. You can actually better represent it in 3D. So we have now VR equipment where you can dive into that. You can look at your favorite phenotype, look which diseases, which genes are around, or you look for your gene of interest, you look which phenotypes are around. That's nice. I mean, we biomedical scientists like images, but the bioinformaticians say it's a database. You know? It doesn't really matter whether you have it as a database or as a 3D picture, but still, I think it's nice. So we work on this cluster of diseases, not because we always wanted to do this, but because I did, like everyone else, hypothesis-driven research, and with some favorite genes and proteins. And then I was ending up in diseases or phenotypes where I had no idea about. We did the knockout mice, we didn't have the expected phenotype, then we sent the mice around, and then all of a sudden we had phenotypes in diabetes, in obesity, in stroke, in Alzheimer's. And I thought, what the hell shall I do with it? I want to work in hypertension. That was my original goal. But then I looked at this poster, all the genes, all the drugs that are currently sort of belonging to my favorite genes and proteins of interest were in this cluster. And then I thought maybe that's not a surprise or not a coincidence. So you can define such clusters and design those networks also independent of genes. For instance, comorbidity. If 2 diseases very often occur in the same patient again and again and again, maybe there are not 2 diseases, maybe there are 2 phenotypes of the same causal mechanism. Or if 2 diseases share the same symptoms, maybe there are not 2 diseases, maybe there are the same disease sharing similar symptoms. So you can construct all kinds of disease networks. None of them is perfect yet, I would say, but from a lot of imperfect networks, you can extrapolate a pretty solid starting point. So we are not ghost drivers, we're not the only ones. We work here. There's Christina Kiel, who works on retinopathies, which clinically, histologically look identical, but she showed that these are 3 distinct molecular mechanisms. So from a molecular point of view, 3 different diseases. Down here is our primary immune diseases. Ellinghaus had a nice paper there, and he actually networked diseases by drugs that work in more than one disease. So it's another way to network diseases. And this is a paper here by Sanchez-Vega and Cell, where they claimed they can define all human cancers by 10 pathways. So more complex tumors have several of these pathways affected, less complex tumors, only 1 or 2. I'll come back to that later. So that's what we're working on. And of course, we have some targets that we found in there. We do not expect that with one mechanism, this in our case, it's related to reactive oxygen species and cyclic GMP initially. We can explain all these phenotypes, but maybe some. So what we then endeavored was to determine how big is the pizza slice in all these disease phenotypes that belongs to this disease phenotype. And then in all of these cases, for all our clinical trials, we have to develop a diagnostic. And we want to move away from multi-omics in every patient. You know, that's unaffordable. Eventually, we have to come down to a simple test that even a GP can order to take a patient with a certain phenotype and then find out what is his or her molecular mechanism that leads to the phenotype, to then give those drugs that actually target that mechanism. Those other gray patients have the same phenotype, but a different mechanism. They need a completely different therapy for their causal mechanism. So in going into more detail, these were the genes that we found associated with this cluster. So a couple of genes related to cyclic GMP signaling and a couple of genes related to reactive oxygen species formation. What geneticists now would do, almost in every bioinformatics paper, they take those genes and then they annotate them to cake pathways, which from a therapeutic point of view, what shall I do with this? These are 400 targets now. Shall I treat 400 targets at the same time? So it's totally inconclusive. Bioinformaticians basically hardly ever validate anything they hypothesize. Plus, these Keck pathways that are used, even Keck says it on their homepage, are hand-drawn maps of pathways. Also, what we learn in biology and so on, these pathways, these are more or less imaginations. Of course, these proteins exist. They do what they are sort of the function that is allocated to them. But these pathways do not exist in this totality anywhere in this purity, anywhere in the cell. They actually lead us away from finding causal mechanisms. And I'll show you some reasons for that. What we actually do is we construct the disease mechanisms in an unbiased way. And our starting point is the interactome. These are all validated protein-protein interactions in a cell. So it's a hairball, but you can click on any interaction. You will find the references. When was this interaction shown by whom? What are the data? And then you take the protein related to those genes in this interactome, you mark them, and you look. In our case, the preferred method is, what are the first neighbors of these genes, of these proteins in the interactome, to get a little bit the surrounding of those proteins. And in our case, we got already in the first iteration, these are the cyclic GMP related genes, and only one of all these ROS related genes that was in close contact with these cyclic GMP-related genes in a protein-protein interaction module, it's called. However, there are as many module construction algorithms in the world as there are bioinformatics groups. Every bioinformatics group wants to have their own method. Since they never validate anything, they mainly focus on generating hypotheses. And there are three major families. In our case, it was pleasing to see that no matter which sort of type of module construction we used, we always ended up with this NOX5 gene as apparently the main sort of interference with the cyclic GMP signaling. So from all these KEGG pathways, the only thing that was relevant for a subset of these disease phenotypes were this area and that area, and they came together in this module. Now you could think that's a pretty mathematical, theoretical approach, but last year there have been a couple of interesting papers to support our view what is actually in future going to be a disease mechanism. I mentioned rare diseases, usually monogenetic. Today, a paper in Cell Mechanism came out which challenges this view. Basically, almost every patient with a monogenetic mutation has a different phenotype to the patient with the same monogenetic mutation. So obviously, in all these rare diseases, there are surrounding proteins and genes involved that modify the phenotype from very severe to almost not detectable. So even monogenetic diseases are not monogenetic. But let's assume for the moment they were. All other diseases are caused by less penetrant, several less penetrant genes, as I showed you. We construct then the signaling module through this first neighbor approach. And of course, lifestyle is a huge factor. We know from diabetes, with proper lifestyle, you can probably prevent like 80% of all cases of obesity and diabetes. It's not sort of, we're not prone to disease with this risk. It's just a risk. In some patients, you can't. Even cancer is not so much different. There you have germline variants, of course, and then in some cancers, like for instance, lung cancer or colon cancer, acquired somatic mutations. But in the end, you will also get gene variants and variations of protein-protein interaction modules. And even infectious diseases are not so different. There, of course, you have a combination of viral proteins or bacterial proteins and host proteins, but they also form protein-protein interaction modules, which, of course, you have to define then experimentally. You can't just use the interactome for that. In the moment, the dogma is a little bit that the way to go for gene therapy, for rare diseases, is gene therapy. This is now softening a little bit. And for all other diseases, what we suggest in future will be to have a network-based approach to disease. So forget one disease, one target, one drug. That's not working because disease mechanisms are little networks, not just one target. And of course, you can more effectively treat a network with 2 or 3 drugs than just rather hammering it at one site with one drug. I'll show you examples of that. In our current clinical trials, here's one example where we used NOX5 as a marker. So this showed the distribution of NOX5 in normotensive patients, and in hypertensive patients, most of them have fairly normal levels, but there is a clear subgroup of patients that have elevated NOX5 levels. And only those are treated. Actually, we prefer biomarkers where we see a bimodal distribution. I personally don't like it very much where you have basically a normal distribution and then artificially like 130 blood pressure. Why 130? Why not 131? Why not 129? You know, that's arbitrary. We actually prefer when there is a mechanistic biomarker, we expect a slight shift in one or the other direction so that you don't have to guess where the patient group probably sits. To actually apply this, we use drug repurposing, which means bringing a drug, introducing a drug for a new therapy that is already on the market. And that's, of course, a very attractive approach because you can basically skip the entire preclinical development. Sometimes regulators want a preclinical validation if, for instance, the benefit-risk ratio is not so optimal. Ideally, you go into a phase 2A trial. At least that's the opinion of the European regulators. And I think it's also an ideal handover point between academic research and industry research. However, clinical professors need to do much more investigator-initiated clinical trials. If we're honest, a lot of hospitals use industry trials as a tertiary income. And that hampers very often clinical scientists if they want to do a cancer trial, but there is a company trial where the hospital gets $30,000 per patient. And then you can't recruit into this trial, but there's always this industry trial competing. In Germany, the situation is particularly bad. There we know that only 1/5 of all trials are investigator-initiated. Only half of those are multi-center. And most shockingly, only 11% are published. So only 1% of all clinical trials are investigator-initiated, multi-center, and published. So it's basically not existing. In France, for instance, 80% of all clinical trials that happen in university hospitals are investigator-initiated, which means the investigator gets the funding, writes the study plan, organizes the monitoring, analyzes the study, and so forth. So this infrastructure you, of course, need for that. I'll show you an application of that. We use this NOx5 inclusion in a hypertensive trial, in resistant hypertension, and we also recently used it in heart failure with preserved ejection fraction trial. Some of you may know that that's a not too infrequent form of heart failure, which does not really respond very well to the current drugs. Here is this cyclogene signaling, here is this NOx5, and apparently in some of these patients, this NOx5 is upregulated, higher than it should be, and that causes apparently an interference with the NO synthase. We don't have to go into the detail here, and you can recouple this uncoupled NO synthase by offering citrulline, which gets much better absorbed than arginine. It together with folate, which regenerates cofactors. It's a bit detailed. You can basically recouple that. You can also combine it down here. You stimulate cyclogene synthesis by another drug that does not release NO like NO donors. So this shows you this network pharmacology approach, twitch at several sites to correct this. And then we expect that this module somehow normalize, maybe not perfectly, but much more to normal. And that trial in HFpEF was actually positive. There was a perfect correlation in the response between NOx5 levels and the therapeutic response in every patient. Then in stroke, the situation is slightly different. It's the same module, but it also shows you that in the same module, different things can happen. There we also have the upregulation of NOx5, which increases your stroke risk. But what also happens in stroke, due to the hypoxia, another gene, NOx5, gets induced, which the normal role is to open up the space between endothelial cells to allow angiogenesis. In ischemia in the brain, that's something you don't want because then the blood-brain barrier opens. And then the overall load with reactive oxygen species is even higher. So then very toxic metabolites from NO and RS form. And so what you want to do here is you want to inhibit, at least for a couple of hours, the NO synthase and also these NADPH oxidases and stimulate that, of course. The problem was there's no NOS and NOx inhibitor on the market. So there you found, however, that you can use an antithyroid drug, PTU, and an antipsychotic, perfenazine, as perfect inhibitors of NO synthase and NADPH oxidase. Why? And that also is a trial that's now finished. Why is that important? Because also forget the dogma, one target, one drug. This is the over 400 FDA approved drugs. And to how many proteins do these drugs, small molecules, actually bind to? There are some drugs that bind really to 1 or 2 proteins, fine. But there are also some drugs that bind to hundreds of proteins. In average, a small molecule that is currently on the market binds to 32 other proteins. Same binding site, same affinity. So terms like beta blocker is completely misleading. It suggests this compound would only bind to beta receptors. No, they bind in average to 32 other proteins. And this is, of course, what you can utilize. The reasons for that is that small molecules don't bind to proteins. They bind to little pockets on proteins. So deeper pockets, shallow pockets, hydrophobic, hydrophilic. But apparently the number of different pockets that we can all have on our proteins is limited. The number in the moment that most structural biologists can agree on, it's an old field, pocketome, another ohm, is 1400. There are only 1400 different pockets on all our proteins for small molecules. Antibodies is a different story, they can bind anywhere, but small molecules, only 1400. That means with 1400 drugs, we will be able to cover all binding pockets on all proteins. Once we develop the 1400 first drug, that's already not necessary anymore. So it shows you there will be an end of drug discovery. We will not need to eternally discover new drugs. And this is what we used in the stroke trial. So one argument you might make is, now you're doing network pharmacology, you use, let's say, 3 drugs, for instance, and they all bind to 31 other proteins. Do we now have to deal with over 90 other targets and a huge amount of side effects? No. And the reason is that when you treat such a module, you get a huge synergy between these drugs. It's not like combination therapy that we do nowadays, a beta blocker, a diuretic, and an ACE inhibitor. They target modules and networks that are completely unrelated. Here, you use 3 drugs that work on the same network, so they are highly synergistic. So we can lower the dose and lower it so much that the individual targets that they may have, for them, the dose is too low. The effect is only generated within this module by this synergy. Here is an application how we are moving now into cancer. That's the Sanchez-Vega paper. They looked at all different cancers. They thought they can define them by 10 different pathways. And then, of course, there are more complex tumors like colon cancer and so forth, or lung cancer, where several modules are affected, and not so complex tumors like thyroid cancer. So to get into this topic, we, of course, focus at the moment on the lower hanging fruits, so low complexity cancer, to basically work ourselves into that. What we don't agree with this paper is this pathway definition. They, of course, as I showed before, annotated genes to take pathways like geneticists do. But when you look in detail at these pathways, they are all interconnected. So it's basically not 10 independent pathways, it's one hairball in the end. So not actionable. So what we do in the thyroid cancer, same thing. We start with validated seed genes. One problem there is that all databases for disease-associated risk disagree. So we have to do systematic review or meta-analysis of the entire literature to get reliable genetic information on these cancers. But then we do what I showed you before for this simpler case where NOX5 was found. Of course, in a tumor, you expect several modules to be affected, not only one, explaining all occasions of this tumor. So you get a very dense PPI network in the beginning. One shortcoming of this interactome is that every protein is there only once. So also a chaperone or a kinase is there only once, but they appear obviously in different contexts. So you take those hub proteins first out to see basically through this, then you can do a community identification. And this is still only a protein-protein interaction network. To interpret it as a pharmacologist or as a biologist, you need to convert this into a signaling module. So the same proteins, but you basically rearrange it in a signaling manner. So ligands, membrane receptor signaling nucleus, if it's in that module. So then it becomes interpretable. The PPI network is not interpretable. You don't even know, do you want to inhibit or activate that target? So these were then in thyroid cancer, all the signaling modules that we found. Of course, no patient is so unlucky that they have all these modules, all these gene variants, and therefore all these modules in their tumor. So we got 4 cells from cancer patients, and we expected that not the patients will differ, and that was indeed the case. So the colors shown now of all the possible modules, these were the modules in color that were affected in this patient. And now we tested this network pharmacology approach in one of those patients. The other tests are ongoing, so these are all unpublished data. So here, this patient has in principle 4 modules. And when we, for instance, test 2 drugs, because of patent, I can't show you the drugs here, in a little module where row A plays a role, but it's only 10 or 15 proteins. It's not the entire row A pathway, as you know them from the textbook. You see, you can reduce the concentration to a level that you have hardly any effect. And when you combine them, the same low doses, you get a super additive effect. Then we looked, what about neighboring modules? And again, suboptimal doses, and if you add them, you get a super additive effect. Even if they're sort of not directly connected, the modules, you get a super additive effect. And then, of course, in the end, in the therapy, we would give the drug combination for all these 4 modules at the same time. And I said, is this all weird mathematics or so? No, there are a couple of really fantastic papers that have come out last year. For instance, in the open cell cartography, they have analyzed how are actually proteins organized within the cell. And what they find is that within the cell, all proteins form little, they call it communities, but it's the same thing. And the size of the community is usually 10, maybe these guys are a little bit larger, between 10 and 20. There are some bigger guys, but the majority of these communities are between 10 and 20 proteins. Exactly what we find from sort of the bioinformatics approach from the interactome. And what they also show is that these communities are connected. So signaling actually does not work in these hand-drawn pathways. It works in little switching modules. So module, they switch to each other, and they are always connected to one protein that appears in the first module and in the second module. So this is how signaling works, profoundly different than we have been taught. And here's another study from Manuela Zaccalo. She works on cyclic AMP signaling. Cyclic AMP is a monster KEGG pathway. You know, gazillions of receptors all go to acyclase, to one pKa, and gazillions of substrates below there. What she has shown is, depending on which G-protein coupled receptor you stimulate at the cell surface with very low concentrations, you get totally different phosphoprotein patterns within the cell. So there is not the cyclic AMP pathway. But depending on what is the G-protein coupled receptor, you have different cyclic AMP signaling events. Of course, pKa and acyclase are in all those modules. Again, they are hub proteins in the interactome, but they have different substrates. It matters what is the input on the top to then see what are the phosphoproteins come out of that. Another thing is, you may have seen are these good cell watercolor pictures where he has actually, from structural biology, microscopy data, and also bioinformatics, drawn how does a cell actually look like. Not like in these nature figures, a receptor is here, a second messenger is formed, it swims over here to the kinase, it phosphorylates some protein, the phosphorylated protein swims up back to the membrane. That's comics. There's not the space in the cell for that to happen. A cell, you can look here, is proteins squished together. Some proteins, of course, may be transported along the cytoskeleton and so forth, but the signaling events can only occur in these small nanodomains. And we have to totally redefine them. Pathways have drawn us into a direction like organ-based medicine. You know, it may have made sense for a while. If you really want to memorize all tyrosine kinase, take KEGG pathways, learn it by heart, but it has nothing to do with reality. And also this one module that I showed you is not only, we have a mix of two KEGG pathways in this module. And that's the final paper that came also last year. Here they stained thousands of cells with antibodies that can mark structural elements in the cell. And then they integrated all these cell images into one. And what came out of that is that what's also wrong, what we often do, we draw a cell and then we put a mitochondrion here and a mitochondrion there. There's a little bit space there. This is how we think a cell looks like. No, it doesn't. All these organelles in the cell are highly concentrated, almost like in organs within the cell. And in this homogeneous area, you see again, there are apparently subdomains. So some call it protein communities, we call it modules. The signaling people call it nanodomains. Is always the same. So what we would suggest, how we should reorganize research, this classical model, you need basic research, you need animal experiments, and then you need clinical trials. We think this model is totally broken. If we really want to cure patients, we need to focus on patients and patient data, clinical data and clinical trials. There is an unbelievable plethora of patient data out there. With those, we have to come to entirely new mechanism-based disease definitions, basically a molecular pathology. The drug repurposing is actually easy. I thought that would be the toughest part. Is actually easy. What we spend most time in the wet lab is to develop assays, simple assays, with which we can sort of identify the patient that has a phenotype and the mechanism. Based on the fact that 60 to 70% of all proteins are expressed in all cells. In the moment, we go for those studies where we can actually detect the disease mechanism in a peripheral blood sample, either plasma, exosomes, or a BAFIB code, Gemisch, because 60% of all proteins will also be in there. So 60% of modules we will be able to detect there. In cancer, it's different. There we go for the whole genome sequencing of this individual tumor, slightly different approach, but in principle, that's our approach. Animal models, only if they are needed from a regulatory point of view, and only if they directly reflect the human mechanism, not just the symptom. And with this, I'm pretty confident that medicine will change, namely to start curing diseases. The risk will remain, but we can cure the disease. Or ideally, if this has been done a couple of times very successfully, very evidence-based, we can probably in a couple of years, everyone will have his personal genome, and then we will identify some risk and then monitor, is the risk manifested in a certain patient, so that we could even start a therapy before the symptoms arise. That would be almost preventive intervention. And with this, the Kondratyev wave will end. So we need to move from organ-based approaches and chronic diseases to a curative system-based approach, bring the entire medicine together again, supported, of course, by decision algorithms, machine learning. We will watch the end of big pharma, not of pharma, but of big pharma, because we will very soon have all the drugs that we need. And we need to move away from single target thinking, move away from these pathways towards modules or protein communities, however you name it, it's always the same thing, and a network pharmacology approach. And this also will be the end of this high NNT problem. Every patient that will be treated in future will benefit from his or her treatment. I think that's it. Thank you.